Hello, everyone. Welcome to the webinar for Finnish Society Bioinformatics Societies. So today we are going to have the speaker, Dr. Jing Tang from Helsinki University. I think this will be the last webinar of this semester. And Jing Tang is currently associated professor at Helsinki University. Actually, he started his PhD there as well, and he has been there for since 2009, and he has been working there for a very long time, and he's starting his professorship like 5 years ago, I guess, and he also received his prestigious ERC starting grant. And today he's going to talk about some computational and experimental methods to tackle the drug resistance in cancer. Actually, his lab is working on this network pharmacology methods for precision medicine. So the stage is yours, Jing. Go ahead.
Thank you. Thank you for the kind invitation to this webinar series. My name is Jing. And today I'm going to give some brief introduction to what the research project of my group. So it's about drug resistance and also a combination of computational and experimental approaches, particularly for the cancer treatment. Before I go to the actual presentation, so this is simply an advertisement. And in the coming months, in August, we'll have a conference in Helsinki called International Symposium on Bioinformatics Research and Applications, so-called ISPRA 2025. So if you are interested, you may go to the website that is listed here for details about registration. And so we have already like keynote speakers determined, and also there will be highly interesting conference program and talks, mainly in bioinformatics research. So I would highly recommend you to go there and take a look. So this is the website of the conference. So today my topic will be mainly about two parts. The first one, I will mainly cover about the background and particularly in the challenges of the precision cancer medicine. And I also talk about the method and the resources that can be utilized for facilitating drug discovery and drug response prediction. In the second part, I will talk more on the combinations. So basically, I will introduce several of the methods that can be used for predicting, testing, and understanding precision drug combinations. And if time allows, I will also share some of the ongoing projects, particularly in drug discovery using AI approaches. So this is so-called 5 pillars of cancer therapies. For the moment, we are actually have a lot of different options for cancer treatment. And also in the recent years, we have the targeted therapy and immunotherapy as active research fields for effective cancer treatment. But if you look at the from a patient point of view, like if you ask a question, so how many patients will benefit from the cancer drugs? Actually, it's just a minority of the patients who can benefit from it, even though the drug has been already approved. So some statistics says that many cancer drugs benefit at most 25% of the cancer patients. So why is that? Because drug resistance problem is a major issue here. And if you look at this clinical trial, and usually that's the basis for a drug to be approved. So here we have a so-called like standard therapy treatment, which is a chemotherapeutic drug. And then we want to show that this new drug is better. So we see the Kaplan-Meier curve for both of the treatment strategies. And you can see, despite that there's an initial response of a new therapy, but in the end, these two curves converge, suggesting that the efficacy of the new drug is not as promising as earlier stage. So this is actually an example of how drug resistance will happen and finally affect the overall efficacy of a drug. So this is a particular example of vemurafenib for treating melanoma, a subtype of skin cancer. So this is also reflected in the cost efficiency of drug discovery. So if you look at the average cost of the drug development, and also the in terms of their long-term sustainability, so that's not actually very cost efficient. And this figure also shows one example of the vemurafenib treatment that was shown earlier on treating this particular patient, and the drug response is very good in the initial stage. For example, within 1 month, like 28 days, you can see probably there's a cure of the cancer, but actually just soon after that, the drug is not working anymore. And at this stage, usually we're lacking of any more options to treat the cancer after relapsed. So this actually brings up the great challenge or like a critical questions to solve precision medicine. And so this is a figure that I like it pretty much, even though the original purpose of the figure was not for showcasing the problem of precision medicine, but I tend to borrow a figure for illustrating the current state of art. Imagine that this is your objective to achieve, but we don't have many like translational approaches to tackle this question. For example, if you go to genomics, normally people can just collect a large amount of individuals and try to sequencing their genomics and to figure out what happened on the genomics for a particular disease. You may also try to develop animal models to understand better the tumor biology and the treatment response. And also, you may utilize the high throughput drug screening techniques for screening a large amount of compounds for their efficacy and potential. But none of them actually are answering the question directly. So this is actually how I started to become interested in this problem, because like 10 years, actually more than 10 years ago, when I first joined at University of Helsinki, so I was working at the FIM. So this is Institute for Molecular Medicine in Finland, which is a subunit of University of Helsinki. So I think we're like forerunners of this precision medicine platform. So far, at that time, actually, we have not utilized this term called digital twins, but nowadays, this concept of digital twins getting more popular. So I think which exactly reflect what we are trying to do, like what we are trying to achieve already 10 years ago. So why it was called digital twins? Because actually we are kind of starting from actual patient and try to get some sample from cancer tissues or from a blood if it is like blood cancer. So we try to then culture these cancer cells ex vivo and run multiple omics profiling as well as the drug sensitivity testing, so that we can create a miniature of the actual patient in a lab environment. And later on, we have the data basically you can consider the genomics, like multi-omics profiling as the genotype of the disease, and then the drug response as the phenotype. So we're trying to establish the relationship between the genotype and the phenotype, utilizing computational approaches. So the outcome of these computational approaches is usually drug response or prediction of the drugs, so that it can be utilized for clinical decision making. For example, when the medical doctors see such a report from the data analysis, then they can have a better understanding of what is the stage of the disease, and also what might be the new compounds that can be used for treating the patient. So in the beginning, we had this platform tested or developed mainly for leukemia, so blood cancer, or multiple myeloma. So they are like hematological malignancies. But more recently, we have also extended the platform into solid tumors, including pancreatic cancer and ovarian cancer. So I think this is a hopefully it's a good summary of these critical questions in precision medicine. So which is best summarized as the four questions. So the first one is asking who might be responding or non-responding to a particular cancer treatment. And then from the pharmacological point of view, we need a better understanding of the targets of the drugs. So the targets of the drugs or the target disease, so they can actually have both perspective as well. So then we also need to understand why patients will develop resistance. So what happened to the patient genomic profiles in a dynamic way, and how this change of the profiles will affect the drug response. So hopefully with all the knowledge that has been accumulated from those data analysis, we should be able to tell which new drug might be potential for improving the efficacy. So this is the vision of myself, also my group. So we are particularly interested in a so-called like predictive model to understand what happened if a compound is given to a particular patient. So also try to bring this precision medicine point of view to make this kind of prediction more individualized. So in following, I will just share some of the examples about how we are developing the computational approaches to answer these questions. So the first question I'd like to highlight is this part, like what are the targets? And this is also a slide about this complexity of the cancer treatment. Because nowadays, because of the resistance problem, which renders a single drug very short term. So that's why we need to also have a combination of the drugs to cover to have the maximum coverage of the treatment for cells with a heterogeneous genetic background. So this is also a challenging problem to rationalize drug combinations because of this possible number of combinations that you can try. And this figure also shows the current trend of the clinical trials, that you will see more and more combination therapies have entered the clinical trials as compared to the single drug clinical trials. But for majority of the combination trials, there's a little rational here, because those are not actually driven by any of the clinical evidence. So this is also a problem of this combination therapy drug discovery, which means that we need a more interactive way or intellectual way to predict what drugs to combine. So this is so the questions can be further decomposed into several questions. For example, in order to rationalize drug combinations, we need to have the data from the patient clinical and molecular profiles, and then these are the questions that are relevant for answering the combination prediction. For example, like what are the disease targets? So in cancer, that's usually suggests that what particular proteins that you can try to affect, try to predict to change the disease etiology. So these are like disease target discovery. And then from a pharmacology point of view, for a particular drug, then we also need to understand what are the proteins that this drug could bind to. And with combinations, then we also need to predict the efficacy and the synergy of the combinations. And in the end, if we want to figure out what is the so why this combination is working, also we need to understand the mechanism of action of such combination. So the first question is about the disease target, and probably you have already been familiar with this database. So that's usually the first database to look at to figure out what are the disease targets. So this is open target database, which is usually like curating the data from the Jiva studies and try to find out what are the particular targets or like proteins that are dysregulated in certain disease populations. However, this drug disease target information is more on the population level, so it's not necessarily specific to any of the individuals. So it's like accumulation of the evidence as the whole population, which may not be true for individual patients. So then how to tackle that? This is the rationale of our computational approaches. As the assumption is that we try to also utilize so-called functional genetic screening data. So what do we mean by that? So functional genetic screening basically is more like a perturbation. So you try to understand what is the function of a gene by perturb it. So this is pretty straightforward nowadays with the technique of CRISPR, for example, or also siRNA. So basically you try to knock out or knock down using the techniques and to see what happens after the perturbation. So if a gene, for example, is very essential, so basically if after knocking out the gene, the cancer cells died, then that means the gene is very important for the survival of the cancer. So that means the essentiality of the gene is pretty big. So if it is essential for cancer survival, but not for the normal cells, then it might be a potential drug target. So that's usually the basis for target-based drug discovery. So you first identify a target, and then you try to identify a small molecule which can have a high binding affinity to the target. So that's usually the process of the drug discovery. So so imagine that you have done this kind of experiment for a particular cancer cell line. So then actually you will have the genetic essentiality profiles for each of the cancer cell lines. So let's give you the data which is more like personalized. So imagine that each cell line is more like a one sample for one particular patient. Then this gene essentiality profiles will be also unique. So this is the data that we rely on. And with this data, we try to identify which gene is more important for cancer survival, which means that it's more essential. And so this is something that I also need to highlight here. Imagine that you have a drug discovery. So basically, you can see that these three perturbations, that is be like sgRNA, which is basically the CRISPR technology, and shRNA is the RNAi, like RNA interference technology, and the drug perturbation. They are actually affecting the cancer genome at different scale, at different level. So they are affecting, for example, the CRISPR affects the DNA, but the shRNA affects the transcriptomics, and then the drug affects the proteins. So basically, they are kind of correlated because they are kind of a central dogma suggesting that the DNA has something dysregulated, which will lead to the change in the proteins. So this is actually how we started this development. We try to compare the gene essentiality profiles of the different technologies. So here in particular, we're interested in a question about the consistency of the CRISPR and shRNA-based essentiality. So they are like two techniques. And then the question is that if both techniques are actually affecting the same gene, despite that they are at the different levels, so one is at the genomics and the other is at the transcriptomics, should we expect that their gene essentiality profiles is also consistent? So we have done this pan-cancer comparison. So in this figure, you can see each row is a cell line, so suggesting a different sample. And then we try to calculate the correlation between CRISPR and shRNA-based essentiality. So to our surprise, actually, the correlation is not as good as what we thought, despite that they are having the same target genes. So you can see the correlation is actually centered around 0, and many of them even have a negative correlation. So that suggests that a very poor consistency was found between CRISPR and the siRNA-based gene essentiality profiles. And this figure also shows that actually this poor consistency doesn't change much irrespective of what kind of genes we're looking at. So whether it can be a housekeeping genes that is usually more stable or more like constitutionally activated, but we don't see improvement of the consistency as compared to the other genes. So this actually the question we're trying to solve. So we ask the question: Can we develop something better, which can also improve the consistency of the gene essentiality profiling? Basically, that's very important because we need a very strong evidence from this kind of data so that we can call it a drug target. So that's why we try to integrate the CRISPR siRNA. So in addition to the functional screening data, we also try to incorporate the molecular profiling data to derive this consensus gene essentiality score called CES. So it's called combined gene essentiality score. So this score is simply a weighted average of the two functional screening values. So basically the value from CRISPR screening and also the values from the shRNA screening, and with a weight alpha, which is based on also the molecular profiles of the given cell lines. So we try to utilize this parameter to adjust the consistency between these two readouts. And then after that, we try to derive a CS score that shows higher consistency compared to a single technique as is shown here. So this is how we show you that it's better. So basically, we can ask a question. So if you have the CES score, and you also have the CRISPR-based and shRNA-based score. So basically, these values here are like a variant of different CRISPR-based or shRNA-based score, like Demeter 1, Demeter 2. There are two versions of the CRISPR-based score, and also like shRNA and other series. So they are like an existing method for characterizing the gene essentiality. So we show that our score has a better prediction accuracy on the important genes. So basically, the ground truth here is that we know some genes already, which are kind of essential for cancer cell. So we use that as a ground truth. So then we try to match our scoring with those ground truths. So that shows the higher efficacy, accuracy to predicting ground truth, and also a stronger correlation between cell line specific or as well as the average essentiality score. So the data has become cleaner and also more robust. So one of the applications of the CES score is that because we can then try to evaluate what are the false positives for each of the individual techniques. For example, we use the CS score to detect the essential genes that are platform dependent. Platform here means that it may be utilizing the CRISPR or RNAi technology. So some of the genes might be kind of very sensitive to the technology rather than the actual biology. So we found out that, for example, like genes which are associated with these pathways tends to be sensitive to the CRISPR screening. That's why we call it as CRISPR sensitive pathways. You can also check what are the pathways that are kind of affected by the shRNA. So imagine you have a gene which is very essential in these pathways. And then if you have this data from the CRISPR, then that usually indicates maybe a false positive, because that doesn't mean that this gene is really essential. Because the reason why it is false positive is that you don't find the same essentiality if you switch to another technique. So that's the rationale why it is false positive. So here you can also check what are the genes that might be biased in the sense that they not necessarily replicate across different technologies. So the other application of this CSS score is that once we have determined this more like a high quality essentiality profile, then we can try to utilize them to identify the cancer type specific essential genes in the framework of a network. So you can see on the right hand side, you have the ovarian nodes are different cell lines colored by the disease subtype. And then the rectangular boxes are those essential genes that are specific to each of the cancer cell lines. So you can see some of the cells, some of the genes are kind of shared by multiple cancer cell lines, suggesting they may represent like a general essential gene. So then we can go and test whether it's truly essential. For example, this is a SRGN, which is shared by leukemia cell lines. So then we validate this essentiality, common essentiality using the survival data from the TCGA database of the cancer patients. So we show that indeed, if you have a higher regulation of SRGN, then the patient usually have a lower survival. So this is also a validation on the breast cancer. So here you will see that this AGR2 is only specific to a particular breast cancer cells. So we also validated that using the breast cancer data. So this is basically the first conclusion, or the first method that we were trying to develop for the cancer drug identification. So we found that actually if you try to combine the essentiality profiles from multiple technologies as well as the molecular profiles, it will enhance the prediction of a cancer drug target. So then we have briefly gone through this first question. Then the second question will be also about the drug target prediction. So this is a paper that was published several years ago, indicating a problem of of target identification for many drugs. So it suggests that actually many drugs have a kind of a miscategorized target profiles. So this is the example that was given in the paper, particularly about this compound called PF. So basically, this compound is already tested in the first in a stage 1 clinical trial. And if you go to the those compound library catalog, so you can check their like putative targets. So actually, it is indicated as a PAK4 in many of the databases or catalogs. So the authors of the manuscripts actually tested this statement. So they basically they ask a question. So for example, if the PAK4 is really the target, then if you knock out this particular target, then the drug is not going to work because of the target is not there. So the drug is not binding to anything else, if this is true. So the authors of the paper decided to do experiments. They knock out the PAK4 and they measure the drug response. So actually they don't see any differences. So the drug is still as effective as before, even though the putative target has been knocked out. They have replicated in multiple cell lines. So suggestion is that it's not only like specific phenomena here. So this experimental results suggest that actually PAK4 may not be the true target. So this is also like a so-called like polypharmacology problem here for many of the compounds. So here it shows the potential drug-target interactions for a drug called dasatinib and also another drug called sunitinib. So you can see that there are not only a handful targets, but actually hundreds of them, which are affected by the compounds. So the complexity of the drug-target interaction should be highlighted much more than before. So that's why we also try to make some prediction about the drug-target interaction. So here is it's so-called like we are trying to utilize the data from gene expression. So the rationale is that as such, so like if you have a drug and a cell, so basically after treating the cell with the drug, you can observe the change of the gene expression. So some genes can be downregulated, some genes can be upregulated. So this profile is usually called the signature, so like gene expression signature. This is also like something like intermediate, so it can be considered as a kind of a intermediate phenotypic changes. And then you will also have the phenotypic changes of the cells, for example, like the percentage inhibition of the drug, so at the different doses. So basically, with these two type of data sets, which can be measured in the lab, so can we then make some prediction of what are the potential targets of the drug? So here it may be like a prediction of the likelihood of a drug target binding here. So this is a question that has been formulated, actually not originally by us, but also rather by Dream Challenge. So this is more like a competition. Like they're trying to ask for the best algorithm to predict a certain biology and machine medicine problems. And it's really a large competition. So you can see the participants of the competition everywhere. So for this particular Dream Challenge, so the task is to predict the drug target using the gene expression signature and drug sensitivity dataset. So then our approach is that we just rely on assumption, trying to make this kind of a linkage between the drugs using different omics data. For example, you have the gene expression signatures. So here I put it here like transcriptomics, and then you also have the gene drug sensitivity, and then you have the drug target interaction data that is known already. So basically, we try to have the data which has these three arms to come up with your training data, and then for the drugs that you don't know what is the target, Then you try to establish the link between the training data and the so-called prediction data, prediction drugs, by establishing the links over the three different data types. So then in the following, I'll just briefly explain what are the different arms of the data. So for example, for this transcriptomics signature, we try to obtain it from the L1000 connectivity map, which is a very large cohort of studies coordinated by the NIH, trying to establish the so-called gene signature, gene expression signature of a compound. So the number of genes here are close to 1,000. So they only focus on this close to 1,000 genes as the biomarker for the drug response. So for the second part of this kind of linkage model, we try to establish a more robust measurements of the drug sensitivity. So this is how we develop this so-called relative inhibition score to represent the drug sensitivity. So in this paper, we have also shown that it actually has higher prediction accuracy compared to the conventional drug response metric, such as the IC50 or AUC score. So then for this particular challenge, we decided to use the drug sensitivity relative inhibition measurement for representing the drug sensitivity. And for drug target signatures, we also try to develop a more a consensus drug target binding affinity score, so-called KIPA. So from the database that has also been developed earlier. So we utilize this KIPA score to represent the drug target interaction. So now that we have the three arms of this model, so then the training will be based on so-called like a correlation between the challenge drug, so the drug that we try to predict, with those drugs that we know already about their target. So the connection of these training drugs and the challenge drugs can be established by using the three different types of data, as shown here. So this is a performance of our method as compared to the other teams. So for this particular dream challenge, we have two sub-challenges. So basically, there are like two sub-tasks of the same problem, or like two evaluation criteria. So first, they want to evaluate how many of the true targets are included in the top 10 of your predicted targets. They also evaluate the performance using more like a comprehensive or systematic evaluation, trying to get the average rank of the true targets in the list of the predicted drug targets. So you can see that our performance has been relatively good, particularly for the sub-challenge number 2. So these purple ones are our performance. Here, the reason why you see 4 columns is that, well, we have different ground truths. So depending on what kind of source of the data. So, or from different databases or from drug bank, this is more binary targets. And for kinome, it's more like a more promiscuous drug target interactions here. So we have different gold standard, and you can see the performance may like be affected by the ground truth as well. So this is also the headline of this Dreaming Challenge results. And this is second conclusion. So basically, we also touch about a bit about the drug target prediction method using a particular data called the signature and also the drug response. So the take-home message is that we also need to do a lot of like cleaning of the data and also like improving the robustness of the experimental measurement. I think that's also the key for improving the prediction accuracy with a more consistent data, including the database curation as well. So this is the second question that has been answered in this puzzle. So like, what are the target problem? So here, I guess I have only like 5 minutes. So I will touch very quickly about the combination prediction. So these are the two questions that I've covered earlier. Now it's come to the third question. So what is the drug combination efficacy and the synergy? And also the mechanism of action of this combination. So that's why we have to utilize a network pharmacology models. Imagine that you have this cancer cell as a black box. So you have this perturbation of the drugs. You can try many drugs. Most of them are like monotherapy, like single drugs. And then you can see what has been the response of the cancer cell, like 0 or 1 here for simplification. So 0 is that the drug is not effective. 1 is that the drug is effective. So we have to infer what is going on inside the black box. So that would be a typical outcome of a network pharmacology models. So this is a method that we have developed based on the network pharmacology, trying to make predictions about what might be the best network structure within the black box. So this is an example of it. So imagine you have the two topologies. Then you can evaluate the fitness of the topologies by evaluating the differences between the prediction and the ground truth. So for example, if you have a model 1 here, then that basically suggests that x1 plus x3 is 1. So what does that mean? That means that if you have x1 and x3 like simultaneously inhibited, then you will see like a complete block of the pathways here. So that is 1 here. In contrast, if you rely on this second model, then x1 plus x3 is not sufficient to block all the pathways because x4 is still active. So that will give you a result of 0 here. So basically, then you try to evaluate your prediction accuracy of like an imaginary case scenario. What if like two drugs, one is hitting here and one is hitting here? So whether your observation is matching with your prediction. So then you can determine the best model, which has highest accuracy to match your experimental data. So this is the example of this network model for breast cancer cell line. So you can see now you can see basically what kind of combinations are more effective. For example, this combination is more effective compared to this one. So then we also go to the experimental validation of this model prediction. So here you can see clearly what are the potential targets. So the ovarian nodes are the targets, and then the drugs are shown here in the boxes, which are hitting these targets. So you can validate the model at 2 levels. One is the like target level, so you can knock out the 2 proteins. And you can also try to conduct an experiment of a combination of the drug by selecting one from here and the other from here. So this result also shows a very good accuracy of our predictions here. So this is like the differences between the high synergy versus low synergy predictions. And further on, we also utilize this dynamic modeling to understand what is the mechanism of action of these two targets. So this ARKB and ZAC are the two kinases that are interacting. So then we try to enrich this interaction with the protein-protein interactions or signaling pathways in a bigger biological context. So we're using this stochastic simulation algorithm to measure the perturbation response if you have like single and also double knockdown. So you can see that with the double knockdown, so the percentage inhibition is much higher than a single knockdown, also confirming our computational predictions. So this is some more further development of this drug combination modeling on actual cancer patient data. So previously it's only on cancer cell lines. But here we extended to different cancer samples from patients directly. And this shows the validation of the result as well. And furthermore, we have make it an even larger cohort data for this network modeling. So this is about the third question. like combination efficacy and synergy. So previously we discussed about this drug combination prediction algorithm. And then it's coming to the question also how to evaluate your drug combination synergy. So like experimentally, you can do this type of typical combination assay where you have two drugs tested at different concentrations. So basically it's more like a dose response matrix or like a checkboard experimental design. So this is your actual experimental data. But in order to evaluate the interactions between the two drugs, actually you need something more like this. So actually it's more like a landscape or like a degree of interactions at this dose matrix. So imagine that you have a like degree of interactions, most of them are like positive, then that suggests that it is a synergistic interaction. If it is like a negative, then it's more like antagonistic. So that means the drug are kind of antagonizing each other. They are trying to decrease the efficacy of the other. So then in order to have this type of quantitative measurements of degree interactions, we also develop different models like HSA, Bliss, and lower additivity. So they are more like reference models to determine what are the response if there's no interaction. And we also have developed our own interaction model called ZIP to give a so-called more consistent evaluation of the degree of interactions in a data set. So details can be also found from this paper here. So in addition to the method developments, we also develop the software to ease the use of the algorithm, also to make it widely applicable for drug discovery researchers. So this is a website you can try to go to search for the tool. And we also have the database that has been developed to curate the existing drug combination data from the literature. So these are the statistics of the data point here. These are also showing the like visualization of the data. This is the website of the portal. You may want to take a look if you want to develop a machine learning-based method for predicting the combinations. And more resources can be also found from the table here. Like we have multiple like R packages for dose response curve fitting, and also a portal for drug repositioning as well. So more details can be found from the link here. Okay, so this is the third conclusion that I tried to make, particularly for drug combination prediction, because of the drug resistance problem. That nowadays we also need to combine drugs of different mechanisms of action. So then we introduce a method called TIMA to predict combination efficacy. And also we utilize the dynamic modeling to understand the mechanism of action of the combinations. Further mathematical models for assessing the degree of synergy has been also introduced and accompanied by several computational tools online and the databases. So I guess I don't have much time, but now I would very little touch upon the existing ongoing project in the group. So basically, we also go to one step further because previously you see that most of the data are from like samples. They are basically a bulk level. But nowadays, with the technology of the single cells, we can go deeper to understand the the changes within individual cancer cells, like the data point here. So then this type of data should be also helpful for drug combination prediction. Imagine that you have found out certain subgroups or subpopulations of the cancer cells. They may respond to a particular compound. Then you just try to combine the compounds which are targeting different populations. So that will also give you the rationale of the drug combination at the single cell level. So this is a more recent paper. It's a combination of a computational as well as experimental approaches. So we are trying to establish this so-called lineage tracing profiles of the resistance. So this figure shows the idea. So basically, it's also related to the concept of digital twins, because here we try to create two copies of the same population. So here this is like an original copy, then we try to double it so that each of the cells will ideally have two copies. And by splitting it into two, like on average, you will see one copy here and the other copy here. So basically, they are more like twins of the same population. So then with these two copies, they can do something differently. For example, for this copy, you can run anti-cancer treatment and see what are the remaining cells. So basically, you can know what color, for example, here, the red color has survived, suggesting that the red cells are more resistant. But if you don't see any of the cells, like this kind of color in the after treatment sample, then suggesting that this cell might be sensitive, so they died easily during the treatment. And then with the single cell RNA sequencing for this sample, you can then identify their genomic profiles or like transcriptomics profiles. Then you can try to relate the drug response and the transcriptomics for the individual cells. So in the end, you will have this type of profiling of the single cell drug sensitivity. So like you have the individual cells, 1, 2, 3, 4, 5, and genes, gene expressions, and then sensitivity. So 0 means that the cells disappeared in the after treatment sample. But here is more like more count of the cell number 3, and even more count for cell number 4. So this is more like a single cell drug sensitivity data, which can be made possible using these techniques called the resist trace. So with this data, we can then derive the signature of the resistance by comparing the pre-resistant cells, so the cells which are survived in the treatment, and then pre-sensitive cells, so cells that are disappearing in the post-treatment sample. So this shows a full change of such a gene signature. Then how to leverage this data for drug prediction? Then we try to also utilize this L1000 that was introduced earlier. So it's like a signature of the drugs. So we try to make a negative correlation between here and here. So this is your resistance signature. This is your like existing L1000. There are like thousands of the drugs with data as such. So we try to have a drug which kind of overcome or like opposite to this resistance signature. And this was categorized by this connectivity score. So the lower the score, the more negative response correlation between the drug and the signature of resistance. So these are the top hits of the drugs for each of the treatment. So these may be the good candidates of the compounds which can be used for combining the drug here. So that will give you a rationale to have effective combinations. So we also tested this method using a drug combination experiment, and also this shows the synergy, like the degree of interactions of the top hits. So validating our method. Okay, so this is the first question. So basically it's a single cell level, and then we try to also provide a rationale of predicting combinations leveraging the single cell lineage tracing data. And also it shows that it could be like a very effective. So of course, this is just very deep of this question. Imagine that we only touch upon this efficacy problem, but all the drugs have the toxicity and the side effects. So then how to evaluate the combination toxicity or side effect, and how to optimize the dosing strategy. So these questions are open. Hopefully with the more research output from these two questions, we can have a better understanding about this rational combination discovery, and hopefully also be extended to some other diseases such as diabetes or schizophrenia or Alzheimer's disease, for example. So with that, I want to stop here, and this acknowledgement. So you can have more information by visiting our website.